you're in the right place if you're looking for the COVID Information Commons Research Webinar, August 2021. And I think we'll be getting started shortly, Lauren, whenever you're ready.
All right, so good morning, everyone. Thank you so much for joining us today. As Florence noted, it's now about noon Eastern, so we'll go ahead and get started with today's webinar. My name is Lauren Close, and I'm the Operations and Communications Manager for the Northeast Big Data Innovation Hub at Columbia University. I'm also a member of the COVID Information Commons project team. The COVID Info Commons, or KIC, is a COVID-19 research collaboration platform brought to you by the Big Data Innovation Hubs and funded by the National Science Foundation's Convergence Accelerator. Every month, the KIC brings together scholars from all over the country to share their research findings in the form of lightning talks. Each scholar will also engage the community directly, answering questions about their work at the end of the presentation session. Today, our Q&A session will be moderated by the Northeast Big Data Innovation Hub's Executive Director, Florence Hudson, and Tia Selkin, who's one of our students here at Columbia University. We also have on our team today Benji Sango, who's another Columbia University student, and Brian Buckley of Boston University. So Brian and Benji are part of our fantastic backend team, and we're so happy that they're helping us organize this event. Thank you to all of you. But before I introduce you to today's fantastic group of speakers, I'd first like to invite Florence to say a few words and tell us a bit more about the KIC.
That sounds wonderful. Thank you so much, Lauren. So welcome, everyone. We're delighted you could join us today. The COVID Information Commons was started about a year ago in July of 2020, funded by an NSF Rapid Award. And what they asked us to do is to provide a portal where you can find all the information available regarding NSF COVID-related awards. And so we created the COVID Info Commons. Since then, we've had 11, this is our 12th monthly webinar. And we've had 720 participants. We've done 66 PI lightning talks, and they're all available on our website. We also had a student paper challenge, and our 3 winners have been invited to participate and present shoulder to shoulder virtually with the other PIs who are funded by NSF and NIH. And today we actually have our 2nd place winner, Samson Chen, from UCSD and now at MIT, getting his master's degree. So we're delighted you could join us. Please use the COVID Info Commons to find information about research and researchers across the entire COVID spectrum, all directorates in NSF. And our goal is to actually add more NSF awards this year and NIH awards so that we can collaborate to try to combat the COVID-19 pandemic today and into the future. And hopefully we can learn so that we can predict and prevent other pandemic issues. So thank you so much for joining us. And I'd like to hand it off to Lauren.
Yeah, thanks so much, Florence. So now I'd like to introduce everyone to today's fantastic roster of speakers. This afternoon, we'll be hearing from professors and faculty, but also students from a range of universities across the country. We're going to be hearing about a bunch of different topics related to COVID-19 and the pandemic. There will be people who are talking about behavioral changes, different computational models. So this is a really exciting lineup for today's presentations. And I'd now like to invite Professor Vedati of East Carolina University to kick us off. I'm gonna stop sharing my screen, and then Ali, it's you're next. You're up.
Sure. Share my slides. Can you see my slides?
Yes.
Okay, great. Thank you for the introduction, Lauren. Good afternoon, everyone, or good morning, depending on where you are. I'll be talking about our NSF-funded project on multi-scale computational modeling of the lung dynamics in COVID-19 patients. This is an interdisciplinary project, and the project team is composed of both engineers and clinicians from College of Engineering and Technology and Brody School of Medicine of East Carolina University. So we all know that the COVID-19 pandemic still surges on, especially with this new Delta variant more recently. And we know that the virus can cause extensive damage and progressive changes to the lungs, among other tissues and organs, particularly in patients with COVID-19 acute respiratory distress syndrome. As you see in this graphic, in the alveoli of the lung, COVID-19 can cause many complex changes such as fibrosis, cell death, and thrombosis formation. Even in healthy lungs, our existing knowledge of the microscale and mesoscale mechanics of the lung is still very rudimentary, in part because of the complex hierarchical structure and mechanical properties of the lung. So patient-specific CT-based computer modeling of the lung in a disease state such as COVID-19 can provide new insights into lung dynamics and into the role of mechanics in lung inflammation. This image is from a recent paper we published in journal Life Sciences, and it shows that damage to the alveoli of the lung at the microscale can propagate and manifest at the macroscale in complex ways. For example, here you can see that in the image on the left from Perman Lab, edema formation in the alveoli can translate into increased tension and stress in the surrounding tissues. And at the larger scale, this can lead to changes in the compliance of the lung, in part due to reduction in the surfactant released, as can be seen in the image on the right from Gaver's group. I would like to also note that the microscale impact of COVID-19 on the aerospace, interstitium, and capillaries are highly interdependent. As you can see in this graphic, damage to each component of the alveoli, such as the septal wall, can lead to damage to the capillaries and vice versa. For more detail, there's a lot of detail I know in this graphic, and some of it is beyond the scope of this presentation in the interest of time. If you are interested in more detail about these progressive changes in lung mechanics, please refer to this recent publication from our group in Life Sciences. Now, the first step in patient-specific in silico modeling of the lung is clinical imaging. In this recent research project, we utilize a 4D CT imaging technique, which includes lung motion tracking via LEDs. And this method can capture 10 CT images or more of the lung during the full breathing cycle and provides more information on lung dynamics compared to a single static CT image. We have imaged 5 patients so far using this technique, and we are using these images to develop patient-specific computer models of the lung. If you are interested in further analyzing this rich imaging data set to answer other research questions, please contact me to discuss collaboration opportunities. You can see my email address here on this slide. Analysis of the infected lungs requires that the lungs are isolated from surrounding tissue in CT images using image segmentation, as you can see here in this image. This is an example of a 4D CT image that we segmented to show where the ground glass regions, ground glass opacities are, nodules and consolidation regions, and the aerospace where each of them are located. And segmenting these COVID lungs can be difficult. There is a lot of artifacts in the images, especially motion artifacts if the patient is coughing, for example. And ground glass opacities, of course, make segmentation more difficult. So we developed an efficient semi-automatic segmentation approach to be able to efficiently segment these images using open source software Slicer. And you can see a representative result here in this slide. We also submitted an abstract on these methods to the BMES 2021 annual meeting. That's the Biomedical Engineering Society annual meeting. And the abstract was recently accepted and will be presented in October. At this point, we have developed both finite element, microscale and macroscale computer models of the lung dynamics, and we are currently coupling the micro and macroscale models and validating these models. And our hope is that these models will advance our mechanistic understanding of lung function in healthy and diseased states and provide a virtual hypothesis testing platform for us and other researchers. These are the two abstracts accepted for presentation at the BMES 2021 annual meeting. I briefly talked about the first one. The second one is focused on better understanding the mechanical behavior of the lung tissue at the microscale and mesoscale, so we can use the appropriate constitutive models and mechanical properties for tissue in our computer models. In this second abstract, we developed a mesoscale lung tissue finite element model and studied the emergent mechanical properties of the tissue based on the available atomic force microscopy measurements. Finally, I would like to acknowledge funding from the NSF, the CMMI division, and the biomechanics and mechanobiology program. Co-investigators, my research team, postdoctoral fellow Anup Pant, and graduate students Elizabeth Dimbad and Shay Middleton. I also thank the webinar organizers for providing this opportunity for me to present my research. And again, if you have any questions or you are interested in collaboration with our group, please reach out to me via my email, which is here on this slide, vadati.a18@ecu.edu. Thank you.
Thank you very much. That was a fantastic and very interesting presentation, Professor. I'm sure our audience has lots of questions for you about your presentation. So as a reminder to the audience, we'll be hosting a Q&A session at the end of all of the presentations. In the interim, if you have a question, you are certainly welcome to submit those questions in the chat. We'll be addressing them individually at the end of all of the presentations. So next, I'd like to introduce our second speaker, Professor Zhenlong Li. Professor, you're welcome to share your presentation and get started.
Can you see my screen? Good. Okay. I think before I'm going to send a chat. So some systems here. All right, I'm going to share, start playing here. Thanks for the introduction, Laurie, and good morning or good afternoon, everyone. And today I'm going to share some of the work that we have been doing using social media data for measuring the human movement during this pandemic year. So this work is funded by NSF, NIH, and also the University of South Carolina. So it's kind of an interdisciplinary project working with other team members in the university and also other parts of the nation. So a brief background about human movement and COVID-19 here. And human movement is among the very important forces that drive the spatial spread of the COVID-19 virus. And this has been confirmed by numerous studies. You can see the publication from Nature Science and from all the other journals. And so funded by NSF, NIH, and the University of South Carolina, from those kind of grants here, we ask a series of questions related to human movement and COVID-19 spread. For example, here, how well are people following the social distancing orders, and how effective is the order for stopping the spread of the virus? So are there any disparities in implementing these orders? And if yes, what are the driving factors, some socioeconomic factors and other kind of disparity pattern that we can find here? And also how to integrate the human mobility into the predictive models, along with, for example, other data sources like vaccination data now. And there are a lot of other questions that we have been trying to ask here. So those questions are actually especially relevant now since we see the spreading of the Delta variant here. And so we take a place-based view, try to think about how we can measure this human mobility using the social media data here. We are using Twitter data here. And so if you look at this diagram here, we have, we have places here like county, like state, like country, right? Different kind of levels of places. And then we have population moving in each place. You can think about social media user, like Twitter user here. And then we can start by looking at those population flows. But since we are interested in geotagged Twitter, for example, and we are interested in data that they have location information, we can first extract and visualize the population flows, give us just an overall view of how this flow changes. And then we can measure the population, further measure the movement, for example, the average movement distances in the county during a specific day. And then further on, we can measure activities between places. So those kind of movement and measurement here is all in the place and time framework. So this is kind of an overall diagram, a place-based view that how we can guide our study to measure the human movement using the Twitter data here, using social media data. And the following, I'm going to show you some results here. Since due to time limitation, I'm not going to give a lot of details about how we get this, but we do have the publications. You can see at each of the slides here, and this result shows us the changes of the cross-day population flows in March 2020. As you can see, the first one is from March 12 to March 13 last year, and then you can see the color, the intensity of the movement is decreasing gradually as we see the order of national kind of emergency, declare of national emergency here. And we can see that population movement actually decreased, that clearly reflected from this map here. And also we can see the similar pattern when you look at the global scale. So we developed an interactive web portal for visualizing this data here. In the link I just shared with you, the first one is about this portal. It's still there, but we still updated that this portal already. But you can still see the time period. In last year, you can click a time here, a date here, and then zoom in, zoom out the map to see how that looks like. And the second one is that we develop a method to estimate the daily number of visitors and residents at a specific geographic scale, for example, the county. For each county, we try to use Twitter data to estimate how many visitors are there, Twitter visitors are there in each county on a specific day here. And then this is the result for 2017 using the method here, kind of validation of our method here. You can see 2017, we know there are total solar eclipse in the US here. And you can see on August 21st, 2017, there is a very clear green belt showing us that there are more Twitter users, more visitors there on that specific day. And even after this solar eclipse, you can still see some you can still see the blue belt here, but much weaker, right? People start to leave that region. And you can see the spatial temporal dynamics of this visitor changes at this county level in this large geographic scale, like the whole nation here. So this is for this using the solar eclipse as a case study to showcase this. And then if we look at using the same method to look at 2020, like last year, this one shows us the county level visitor changes before and after national emergency declaration in March. So you can see this is a Saturday on March 7th last year. And again, the blue color indicates the green color indicates there are more users, more Twitter visitors, and then the orange indicates there are less Twitter visitors. So on Saturday, normally on the weekend, there are a lot of movement, a lot of travels. And then, but on this Saturday, which is March 28th, you can see the movement had been decreased a lot. There we can see a kind of a lot of brown color, which means a lot of people don't travel and stay at home in their own county at least. So we also have a publication for this one. If you're interested, you can look at the method in more detail and other kind of application here. And we further developed a scalable platform for kind of extracting, analyzing, and also sharing this kind of human mobility data that we get from Twitter and also from other big data sources here. And we combine this kind of billion of mobility and we combine with ODT data model, which is origin, destination, time data model. It's a data cube. And then combine that with HPC, high performance computing. And then we are able to quickly query and visualize and aggregate the flows at different geographic scales and for different applications here. And here, this map shows you the human mobility patterns at different kind of geographic levels. You can see the country level and county level, and also the sensor tract level. You can see the daily trends revealed from this human mobility by using this system here. And finally, we developed the ODT Flow Explorer, which is this online interactive web portal. I also shared this link with you already. You can go there to explore the human mobility at different geographic scale, like tract, county, country, state, and for the whole world. There are some scale that we don't have that for other countries. And also you can visualize, download the data and for your own research. So the portal now has been visited by thousands of visitors, and we have served over 1.8 billion flow extractions from over the world, and also used by other researchers in their own research. So if you are interested, feel free to try this out, and I already have a link there. So that's it. Thank you. If anyone interested in working or in looking at more detail about data that we produce, feel free to contact me. I have my email address at the beginning of here. All right, thank you.
Thank you so much. That was really fantastic, and we appreciate you sharing your research with us. I'd like to now hand it over to Deborah Solon, who is coming to us from Arizona State University. Please go ahead and share your screen.
Thanks so much. Here we go. So I'm an associate professor here at the School of Geographical Sciences and Urban Planning at ASU. And together with colleagues at the University of Illinois at Chicago and folks here, we have been working on trying to understand behavioral changes in households between, before the pandemic to during the pandemic, and also how people expect any behavioral changes that they might have experienced during the pandemic to perhaps persist even after the pandemic is no longer a threat. So that's what I'll be talking about today. We were funded through a RAPID grant through the CMMI program at NSF, and a few other places as well have helped support this work. So COVID-19 has required most of us to really make big changes to our daily lives, and our research is trying to understand both what those changes were, how large they were, and also, but perhaps more importantly, will some of these new behaviors really stick? So we conducted a national survey. The survey we call the COVID Future Survey, and you can learn a lot more about our work at our website, www.covidfuture.org. This is just to show you that I'm obviously not alone. I have working with a large research team. This doesn't even actually, we've added more team members recently, so this is not even fully representative of the group. It's a big effort. The survey we've collected, wave one of the full wave one of the survey, it's a multi-wave survey. The full wave one was nearly 10,000 respondents from across the US. Today, I'll be reporting results on a subset of those people, a bit over 7,000 of them. So the survey sections, we're transportation researchers, most of us. And so we were interested in how this pandemic has affected transportation related behaviors. Actually, I was quite interested in the previous presentation, thinking about movement, But anyhow, our survey looks at how people are accessing these different activities, employment, working and studying, shopping and dining, how they conduct their transportation on a daily basis during the pandemic, pre-pandemic, and expectations for the future. The survey took between 15 and 30 minutes. Wave one was implemented starting in April of 2020 and went through October. The data I'll be presenting today is really more from the July through October period. That sample size was about 9,000. We've actually collected an additional around 1,000 since then. Wave two was implemented from November to April. About 3,000 people responded to that. I'll just make a couple comments on what we found in wave two today, but I don't have any formal findings on that. Wave three, we're going to be implementing in about a month, starting mid-September to mid-October. And we're currently applying for funding to conduct 2 or 3 additional survey waves. We had originally imagined that 1 year would be enough time to, or maybe 1.5 years would be enough time to really understand the effect, during the pandemic and post-pandemic, but obviously that has not unfortunately happened. And so to understand the more long-term impact on people's behavior, we really need to continue following folks into the future. Just to give you an idea of where our respondents came from, they're from all over the US, although there is an overrepresentation in Arizona, as you can see. We to account for regional differences and also differences between our sample and the US population as a whole, we do use a weighting scheme, and these are the main variables we weight on, plus region of the country. More details about a lot of the results I'll be presenting in a minute are available in our recently published brief report in the Proceedings of the National Academy of Sciences, and I'll put a link to that. It's open access, so I'll put a link in the chat after this presentation. So a couple of things. One of the big questions we had was really, would you like to continue any of the things that you're doing now after COVID-19 is no longer a threat? And this is a pie chart just showing how people answered that question. And we were maybe not surprised, but interested to see that the vast majority said either yes or maybe to this question. So there are definitely some things that people are experiencing during this crisis time that they find that they like and that they might want to continue. And so here's just a chart showing what some of those things were. And the top thing is working for WFH means works from home, working from home at least some of the time. But you can see some of these other items are perhaps some of the things you've been enjoying during the pandemic: spending more time with family, taking more walks, spending less money, cooking a bit more, etc. So just honing in on a couple of these main results. So working from home. Our data forecast not only that we've seen a large increase in working from home, but they forecast a large continuation of increased work from home. So you can see here the pre-COVID actual percentage was about 23% of people work from home at least a few times a month. So these aren't people that work from home every day, but regularly and somewhat frequently. So then, and that has gone up to over 40% during COVID period, and people expect to continue this. Now, this is a little misleading that there isn't that much of a drop here, because these people are working from home mostly almost every day, whereas this percentage is more of an indication of like a couple times a week is more of what people are generally saying. But in any case, people are planning to continue this behavior. This chart shows a little bit more detail on that, but I don't think I have time to go into it that much more. Looking at a second kind of related big behavior that we're very interested in is car commuting. So we see that our data forecasts a related decrease in car commuting, perhaps, which is, if people are working from home at least a couple of days a week, you can imagine that this would happen, right? They're commuting a couple of days less. So that's what this is reflecting. We asked a lot of our questions and about what they expect in the future, and then we asked why. And so this is an example of kind of the type of question we were asking, looking at, if people said they anticipated to be traveling less by air after COVID-19 was no longer a threat, we said, well, why? And gave them a bunch of possible reasons that they could select from. And then we divided those reasons into things that were like new realizations, like I realized I could conduct my business meetings using video conferencing, or pandemic related reasons, which were more like, oh, I don't want to fly because I'm worried about getting sick, right? So we think that these new realizations might be more long lasting than the pandemic related reasons for a lot of people. But in any case, what we can see here is that the blue, which is the new realization, is the darker blue. The things that were sort of neutral is the medium blue, and then the light blue is the pandemic related reasons that we think might disappear if COVID-19 really does become not a threat. But in any case, we can see on the business side, there's a lot of people saying they're going to be flying less. Not so much on the personal flying side, but a little bit. And the last, well, maybe not the last, one of the last things I want to talk about was mentioning that one thing, on the earlier slide, I was saying, highlighting things that people said they wanted to keep from the pandemic time period. And one of the things was taking more walks. And that's reflected in another aspect of the survey where we asked people, how often they wanted to use different modes of transportation. And a lot of people said that they wanted to walk more. The next slide shows a little more detail on this. And then I think this is really the last thing I was going to talk about, which is dining and shopping. So we asked about how many times people dined in restaurants pre-COVID and how many times they expected to dine in restaurants, on a weekly or monthly basis post-COVID. And you can see that these frequent diners, the people who said they dine in restaurants a few times a week or more, are really expected to drop. And that's something that I think is likely of concern to the restaurant industry. And so not only can you find more information about our survey and our publications at covidfuture.org, but there's also a link to our data, which is freely available for download. Right now, Wave 1 is available. We'll be posting Wave 2 in the coming weeks, and Wave 3 once we've collected it and cleaned it and weighted it. So thanks so much, and let me know, and look forward to the conversation later.
Oh, thank you so much, Deborah. And it's really interesting to hear that there's already like a little bit of a dialogue and an overlap between some of the work you're doing and some of Zhenlong, who went right before you did. So that's the whole point of the KIC webinar, and we're so glad to see those connections happening in real time. I just want to remind the audience, if you have any questions for any of our speakers, go ahead and share those in the chat, or hang on to them if you want to ask them live at the end of all the presentations. Next up, we have Professor Sarah Stahl, who's from the University of Pittsburgh. Sarah, go right ahead.
Thank you, Lauren. All right, share my slides. All right. Hello, everyone. Like Lauren said, my name is Sarah Stahl. I'm from the University of Pittsburgh, and I'm an assistant professor of psychiatry, and I do research on bereavement and grief, depression and aging. And so today I'm going to talk about bereavement and the COVID-19 pandemic in older adults. And my research is funded by the National Institutes of Mental Health, which is one of the 27 institutes of NIH. And the stuff that I'm going to talk about today was funded by a COVID supplement to my R01 to really try to identify and enroll bereaved elders who experience the loss of someone to COVID-19. So millions of Americans have lost a close family member to COVID-19, and researchers have found that for every COVID-19 death, that this corresponds to about 10 individuals who have lost a grandparent, a parent, a sibling, a spouse, or a child. And a lot of people are suffering from these sudden and unexpected deaths of their loved one. And so we think that this suffering may be associated with long-term health outcomes. So what are the differences between bereavement and grief? Because I'm going to be talking about those two terms a lot throughout this talk. So bereavement refers to the time or the window when a person experiences sadness after losing a loved one. And grief is how a person reacts to the loss of a loved one. So this includes the entire emotional process of coping with the loss, and it can last a really long time. And what we know from several decades of bereavement research is that even though this experience is very painful, most people adapt to the loss of a loved one and learn to live their life without their loved one. However, we think that with all of these special circumstances surrounding COVID-19 death and the inability to mourn and be with family members and likely be socially isolated, that individuals' bereavement and grief trajectories are going to last a little bit longer and possibly be a little bit more difficult. So loss during COVID-19 can be particularly challenging. So I focus on older bereaved spouses. With this study, we're trying to recruit older spouses who lost their spouse or life partner to COVID-19. So spousal loss to COVID-19 can be particularly challenging because a lot of older spouses were likely socially isolated while trying to provide care to their physically declining spouse, which then in turn created a very exhausting caregiving situation. And then after the death, we know that there are a lot of restrictions placed on mourning rituals, which inhibits individuals to grieve. So people who are bereaved by COVID-19 compared to those individuals who have died from other causes, whether it be cancer, dementia, or cardiovascular disease, we think that older spouses bereaved by COVID-19 are more susceptible to negative health outcomes. including depression, anxiety, loneliness, and something in particular that I'll describe in detail, which we call complicated or prolonged grief disorder. So prolonged grief disorder is a syndrome that's characterized by persistent and pervasive yearning, longing and preoccupying thoughts and memories of the deceased. And this yearning causes significant distress or impairment in functioning and lasts at least 6 months and exceeds the time frame that's typically expected by any social, cultural, or religious norms. And prolonged grief disorder was recently added as a new diagnosis to the ICD, and it's likely going to be included in the DSM-5. So there are a lot of risk factors that individuals are experiencing during COVID-19 that may increase the prevalence or prolonged grief disorder. So a lot of the circumstances, contexts, and consequences of death will likely elevate prolonged grief disorder in the coming months. So circumstances like sudden and unexpected deaths, people were dying alone, there were restrictions on visiting policies, and so families were unable to see their dying family member, they were unable to say goodbye, and then there were even instances where family members were not sure where, when they were able to pick up the body or what kind of mourning rituals they were able to engage in. So a lot of the physical distancing policies affected funerals, burials, rituals, and any other kind of support services for the grievers. And even support services, from local hospice organizations had to change from in-person to virtual services. And so people are having a difficulty grieving the loss of loved ones. So as bereavement researchers, we feel that there's an urgent need to educate others on grief and the way that the pandemic has affected our ability to grieve. And we think that family members should try to monitor their bereaved loved ones and look out for things that may signal prolonged grief disorder. So certain thoughts like excessive avoidance, reminders of the loss, social isolation, and any disruptions in routine health behaviors, like their sleeping, eating, or exercising behaviors, may signal prolonged grief disorder. So our work with bereaved adults, we have a efficacy trial that's funded by the NIH, where we are comparing 2 groups. So one group uses digital monitoring to self-monitor 3 health behaviors. So we focus on sleep, meals, and physical activity. And during COVID-19, we transitioned everything to a digital protocol. So everything is online, and we give people feedback about their health behaviors because we're really trying to focus on healthy behavioral routines. And we will compare that group to our control group. And over time, we think that by focusing on the routine of healthy lifestyle choices, that we will be able to reduce depression symptoms, but we're also tracking other important bereavement adjustment outcomes like complicated grief, anxiety, post-traumatic stress. So like I said, our intervention is a digital health intervention for spousally bereaved older adults, and we're really focusing on the timing and regularity of health behaviors to promote a healthy circadian rhythm. And because everything's virtual, our participants, use a web-based platform to self-monitor their health behaviors, and they also receive personalized feedback via this lifestyle log that we've developed. And all of this is, with the goal of trying to help older spouses engage in a regular routine of sleep, meals, and activity. So enrolling participants has been challenging due to COVID-19, but we've been more successful over the last several months and noticed that about half of our participants were bereaved by COVID-19. And what's interesting to us is that our COVID bereaved group, when we compare them to our non-COVID bereaved group, that they're very similar in sociodemographics. They're primarily older women in their 70s. They're recently bereaved. They're only bereaved about 4 months when they contact us. They're both similar in terms of their depression and anxiety symptoms, but what's different are their scores on the inventory of complicated grief. So this is a scale that measures complicated or prolonged grief symptoms. And those that are bereaved by COVID-19, we can see in yellow at the bottom, their score is much higher. So it's around 25, which is right at the threshold of signaling to clinicians that they may need clinical care, particularly at 6 or 12 months following loss. And those that are bereaved by other causes of death, their ICG score is a lot lower, around the 17. So we are seeing that our COVID bereaved spouses are likely suffering differently and more intensely than those bereaved by other causes of death. So what did we learn from talking to our bereaved participants? So loss due to COVID is really distressing, and it's potentially predisposing surviving spouses to adverse health consequences. And we really want to try to work with our local community health partners to educate each other on recognition and treatment of prolonged grief disorder. And we think that there should be a lot of public health efforts to identify and aid COVID bereaved adults using telehealth approaches. So if you or someone you know has experienced the death of a spouse or life partner, or want to learn more information about bereavement during COVID-19, you can contact us or email me. And thank you for listening.
Thank you so much, Sarah. That's a difficult topic, but a very interesting one and certainly important to study. I really appreciate you sharing your insights. I'd like to now introduce our next speaker, Professor Tabeba Zar of the University of Utah.
Masood, I think you're muted. I can't hear you.
Sorry.
That's okay. We can hear you now. Thank you.
Thank you very much for the opportunity, and thanks for the introduction. My talk is slightly different than the ones we have seen so far. It is about developing some electronic sensors to detect COVID-19 from the environment and from saliva and bodily fluids. Dr. Elizabeth Middleton from University Hospitals of University of Utah has provided the saliva samples, and without her help, this wouldn't have been possible. And this work is partially supported by an NSF CMMI RAPID grant. So as I mentioned, the objective was to develop a sensor that is very fast. So in order to be able to do that, the idea came across to develop a sensor that detects the whole virus and not decompose it to its constituents like proteins, RNAs, and whatnot. And we wanted to be able to detect these viruses in less than 5 minutes. So the sensor that I'm describing today does it in about a minute. And also, it was desirable to have reusable sensors for a resource-limited area so that we can reset the sensor and reuse it. Although there are some issues regarding contamination and cross-contaminations that prevents this from happening readily, unless you have some elaborate techniques to reset them without contaminating others. So if anybody's interested in the stuff that I'm talking about, I sent in the chat a link to all the news clips and stuff that cover this sensor development. And also Dr. Aaron Duffy from our commercialization office can be contacted for commercialization discussions. There are 5 companies that have started commercializing this. So saliva is actually pretty complex fluid. 99% of is water. That 1% remaining has lots of nanoparticles in it, and I didn't know that before I started. Like most of the stuff that we do, there are nanoparticles that are completely different size than COVID-19. They're okay because they don't really contribute to the signal as long as they are much smaller than COVID-19 or much larger. So COVID-19, it takes different sizes. In saliva, it's about 125 nanometer in diameter. And directly competing with it are the exosomes in saliva. They're about 100 nanometer in diameter, so similar in size, and they're also spherical, but they do not have any spiking proteins that we are familiar with in COVID-19. So our sensor basically relies on three different things. This is the schematic of the sensor structure. These are optical microscope images of the sensor. Essentially, there are two electrodes that are separated from each other by a distance corresponding to the size of the COVID. And the bottom gold electrode and the top gold electrode, they have surface molecules on them that are called aptamers. These are single-strand DNA synthetically designed to bind with these spiking proteins on the surface of the COVID-19. And they bind with S1 and S2. These are surface type 1 proteins and surface type 2 proteins. So size is important. If it is too small or if it is too large, it won't really fit the sensor space in there. The spiking proteins are important. If they don't have it, you can wipe them out without too much difficulty. And also the electrical properties of these viruses are also important because we measure the impedance of this junction, where you can call it the capacitor or whatever you want to call it. And if it is not COVID-19, then the impedance that you would measure would be different. So these three different things should come together for us to recognize something as the virus that we are interested in. COVID-19 is also electrically charged. This is well known about viruses. In different pH environment, they acquire different residual charge. In the case of COVID-19 and saliva, it is slightly negatively charged. You can see that if you perform these current versus voltage measurements very carefully with electrodes, one side functionalized with aptamer and the other side not, you can see that this asymmetry. And to explain this asymmetry, you have to assume that COVID is slightly negatively charged. We take advantage of that and put positive charge at the bottom electrode in our sensor in order to attract actively the virus to the sensor. So once you have the sensor in place and all the aptamers and so on and so forth, you can choose different ways of measuring the response of the sensor to the presence of the COVID-19. You can do current versus voltage measurement. Those are DC measurements, or you can look at the capacitance and resistance of the sensor and then try to see if the presence of COVID-19 significantly contributes to the behavior of capacitance and resistance over certain range of frequencies. And at 5 kilohertz, the difference is largest between saliva that's infected and saliva that's not infected, that are deposited on the sensor. Of course, the inductor capacitance resistance measurement units are 50K or so, and you can't really do that in a handheld device that you want to basically give people something like this to carry with themselves to detect the COVID in the environment. And we replaced all those equipment with a microprocessor that applies these square pulses. And then we developed a technique to just look at the response of the sensor to this output voltage that you have. And we have a reference capacitance that is comparable to the capacitance of the sensor. And if the V out kind of looks like the red one that I show in there, then we decide that it is the saliva is infected. And you can see the response of the negative and positive. There are a few samples that are kind of in the no man's land. And those are the false positives and false negatives that we account for. Now, the system is a standalone. There is a LED that turns red when the saliva is infected or it remains green. And this system is also can be paired with your smartphone, and the output of the sensor can be displayed. There is also a mapping capability that enables you to see where in the world the sensor has been used. And if there are infections detected, then there is this, COVID-19 schematic or whatever cartoon shows up. And there was one in Provo in in Utah. And you can see that it is being tested in many different places, Japan and Thailand and all over the world. Now, detecting things from airborne kind of version of detecting the airborne version of the COVID-19 is a little bit more challenging. The false positivity rates and negativity rates are slightly higher because when you do it from airborne samples, there are soot particles in the air and so on and so forth that are similar in size, and one has to be careful about that. But still, we can detect it from airborne samples without too much difficulty, although it needs more development to be able so that we can use it in the air rather than putting some saliva samples on it. So these are some of the statistics or the characteristics of the sensor. Our false positivity and negativity rates are between 4 and 10. It is better on the 4% side when you do it in the laboratory environment, again, because the sensor interacts with the environment, and less stuff you have in the air, better it works. And it detects the variants, but if the variants have S1 and S2 proteins that are more than few percent different than the original one, then we have to change our aptamers. These are synthetically produced, so they're easy to get and employ and use it in our sensor. Our sensor system also has an oximeter and thermometer and walk sensor, so it can enable you to look at the toxic gases in the air, CO, CO2, stuff like that, that are also become larger in infected people. That's basically it. Thank you very much.
Well, thank you very much. That was really interesting. And it's, like you said, a different topic than what we've heard so far. So we're just adding to the cornucopia of interesting conversations today.
Thank you.
I'd like to introduce our final speaker today, who's Samson Chian. Samson is one of our 3 winners of the inaugural KIC Undergraduate Student Paper Challenge, which was conducted earlier this spring. So we welcome Samson, and we're very excited to share his research as an emerging scholar. So Samson, please go ahead and take it away.
Thank you, Lauren. So today I'll be doing a presentation, which is also slightly different from the previous ones, about generating explanations for machine learning predictions on viral and bacterial pneumonia. And so the overarching goal of this research is to take various chest x-rays of patients who have bacteria and viral pneumonia, as well as healthy patients, and build a machine learning model to classify between these patients using these images. And so is it possible to build a machine learning model that can accurately identify these different classes, but also interpret this machine learning model in order to understand where the model is looking at when it's generating its predictions. And so this introduces the idea of using explainability algorithms, also known as explainable AI, to analyze a model and the data it's predicting on in order to understand exactly where the model is looking at. And the ultimate goal is to see if this type of framework can help radiologists in their work in diagnosing different patients. So a quick overview of explainable AI. There are various types of explainable AI algorithms, more like a family branch of different types of methods. For this research particularly, we'll be focusing on post hoc algorithms, meaning algorithms that analyze much complicated models after they're trained on the data instead of during the process or before. And the benefit of this is you're able to use more complex models, such as deeper convolutional neural networks, in order to learn the features in the x-rays. And there's a trade-off between interpretability and accuracy sometimes. And so deeper and more complex models are harder to interpret than simpler ones. So these algorithms provide a way to understand these more complex models. So the first type of algorithm is known as LRP, layer-wise relevance propagation. And this is a method that I'll be talking more about a bit later, but this essentially looks into the model's layers and weights and understands what pixels in the image contribute most to activating a model's internal structure. The second one is something known as LIME, and this method is model agnostic, meaning it doesn't depend on what type of model you use. And this method differs from LRP in that it starts from the data first rather than the model, and it looks at individual subsections of the data to find what regions in the image contribute most to a model's prediction. And then the next one is called GradCam, which is similar to LRP sort of, but instead of looking at each layers and each weights, it takes a look at the convolution layers in the model, and then it takes the gradients that when you fit an image, the gradients of the last convolution layer, and then it produces sort of like an activation map of the gradients. And the last type of algorithm is sort of more of a novel algorithm known as contrasted LRP, which is a modification of the regular LRP, but it takes the relevances and then it differs between different classes such as viral and bacterial pneumonia. So you can visualize the difference between the classes more. So building a convolutional neural network to identify the features in these type of patients requires a complex model structure, such as VGG16 and ResNet-50, which are two state-of-the-art models that do a very good job in image classification. And as you can see from the model structures, it's a very deep convolutional neural network, which is able to pick up on many features in the images. And so these two models give some of the best results in classifying between the three different classes of patients. And so the X-ray images were all collected from Mendeley data, and a bunch of image pre-processing is required on the images to fit it into the model. So making sure that there's a similar amount of image examples from all different type of classes, and then doing some pre-processing on the images to fit it into the model. And here's like a image of just an overview of the model performance during training. So on the left, you have the accuracy plot for the training set and validation set of images. And as you train the model more and more, each iteration, each epoch, the accuracy grows more. And then the loss, which is what we're trying to minimize on the right side, is steadily decreasing. So this shows that our model is picking up on the features in the data, the images that we're feeding it, and it's doing a good job in classifying between the chest X-rays. So here's just more analysis on the model built, and it's very important to have an accurate model in order to identify or examine what the model is looking at each image. So you can see a confusion matrix on the left, which shows what the model is confusing, and in general is doing a very good job between the three classes, besides distinguishing between viral and bacterial pneumonia, which is doing it's getting confused on a few examples. And so it's important to run these explainability algorithms to understand what the model is looking at. So here's a brief overview of LRP, layer-wise relevance propagation again. And essentially what this does is you have your neural network with each layer, hidden layer inside, and it propagates your image from the output layer backwards. So instead of forward propagating, it propagates the image backwards, and then you compute relevance scores at each neuron, which is these circles right here. And these relevance scores represent how important each pixel in the image is to a prediction that the model makes. And so here you can take a look. On the right is an example of LRP. And as you can see, it produces sort of like a heat map based on the individual pixels in the image. And that shows how important each pixel in the image is contributing to the model's prediction of the class. And then on the left, you can see comparison of LIME, which is more of a region-based method that takes regions in the image and then finds which regions are most important to making a model prediction. And then Here's more examples of Lime for the three different classes. What Lime does, it doesn't specifically go into each layer of the model to examine the weights, but instead it takes subregions chosen specifically on the image and runs on the model to determine which of those subregions is most important to the model's prediction, and highlights those specific regions. And then another type of algorithm that I discussed was called GradCam. And GradCam essentially is an algorithm sort of like LRP, but instead it examines the gradients of the convolutional layers in your model. And so this method also takes an image and then feeds it in to the model, and then produces an activation map, which is a heat map that represents all the gradients in a convolutional layer. And that picks up where exactly the model is looking at. So here are some examples of GradCam instead. It's sort of like a combination between LRP and Lime kind of. It produces a heat map of a specific region on the image. And you can see what the model is focusing on and what regions it's not focusing on. And the last variation, which is known as contrastive LRP, is a slight modification to the original LRP method, but instead it takes the relevance scores and applies a modification that distinguishes the relevance between various types of classes. And so you can identify or distinguish between viral and bacterial pneumonia a lot more clear using these different types of relevance scores. And so it's very important overall to look and compare these various types of explainability methods in order to understand what your model is specifically looking at. And so there are many types of algorithms that you can use. But in order to get a thorough understanding of what your model has learned, what your model is looking at when it's making predictions, it's important to compare these types of methods and while you're building an accurate model to help patients. And just some ongoing research right now. Explainable AI is a continuously growing field. More algorithms are being developed every day, and more data is being collected to build more accurate models. And different types of data, such as CT scans, can also help with radiologists' work in diagnosing patients. So running these algorithms on different types of models built to examine which model has learned the data more accurately than others. And yeah, thank you so much for listening to my presentation. And I would like to acknowledge Dr. Michael Bazzani, who's also on the call right now, for all of his help in conducting this research, collecting data, and implementing these algorithms to identify pneumonia. And thank you. This is my email if you want to contact me.
Thank you so much, Samson. It's really exciting to see your research evolve, and we'll be, watching your career and your scientific research with interest. We really appreciate you sharing your work with us, and congratulations again on winning our student paper challenge. I want to also thank all of the researchers who shared their work with us today. This has been really interesting. Each of your presentations provides some unique insights into the current COVID-19 pandemic, and we appreciate your time. I want to also note that each of our speakers' presentations will be made available on our website, covidinfocommons.net, later this week, so keep an eye out for that. And now I'm going to briefly share my screen, okay, and just announce to this group, there you go, announce to the group our upcoming webinar, because we do this every month. So our next webinar will take place on September 22nd from 2:30 till 4 p.m. You can register to attend this event on our website. I've provided the link in this slide, so pencil that into your calendar, and we hope you'll join us then. An additional item that I want to share with the group before we head into the Q&A session is about a new NIH RFI, and I want to hand it over to Florence and our guest Orlando Lopez, who's joining us from the NIH, to say a bit more about this opportunity.
Thank you so much, Lauren, and thank you to all of our presenters. Great job. And Samson, we love seeing the students in the mix, so thank you. And thank you, Dr. Pisani, for mentoring him as well through the program and the process. So now I'd like to invite my colleague and friend, Orlando Lopez, who's from NIH. He is very involved in this new RFI on input on technologies and approaches that can identify individuals susceptible to developing post-acute sequelae of SARS-CoV-2 infection, or PASC. And there's an RFI that NIH has, and the input is due by August 27th. So since this is a COVID research crowd, we thought it would be a great opportunity for him to talk a little bit about it. So Orlando, if you could introduce yourself a little bit more and the opportunity for people to participate, we would love it. And thanks for joining us today.
Oh, thank you so much for the gracious opportunity to participate in this event, and of course, to share this request for information with the group. So yeah, so I'm Orlando Lopez. I am a program officer with the National Institute of Health, specifically with the National Institute of Dental and Craniofacial Research, and I've been part of the initiative that started last year around rapid acceleration of diagnostics, which it's referred to as RADx. That was essentially supported directly from Congress and charged with really ramping up the capability for testing around COVID. At the start of the pandemic. And so this RADx initiative was being supported. I think we got about like a $1.5 billion in funding, specifically to address a lot of these issues. So there's a number of different program areas that were or have been encompassed through the RADx initiative. The first one started as RADx Tech, and that one really aimed to get technologies that had some really great potential, maybe were in the development stage, but were like really almost ready for scale-up manufacturing and really getting to the commercialization pipeline. And so that was funded. I think over 1,000 different technologies went through that process in a Shark Tank-like version. Significant infrastructure has been in place, a lot of public-private partnership infrastructure to be able to vet a lot of these technologies and really push this forward. There is a number of other program areas. Altogether, I think there's 4 different RADx programs. I've been involved with the baby version of it, which is the RADx Radical. And as part of that cohort, we've been pushing for the development of very novel technologies that aim to identify the SARS-CoV-2 virus in different ways, or also are able to diagnose or detect the presentation of the disease in different formats. So as part of that initiative, I've been leading the Novel Biosensing initiative, which is a cohort portfolio of about 20 projects that have been funded across academia and small business. And we're looking at a number of very exciting technologies that encompass a number of different detection schemes to identify the virus from the oral cavity. So like some examples will be detection of the virus directly from exhaled breath, so similar to like a breathalyzer type technology, but we're looking to develop not just these technologies to address this particular pandemia of COVID, but potentially establish new platforms that could address future pandemias and future health challenges. So we can envision the detection of different biomarkers and biosignatures from breath, to encompass a number of different capabilities that can help us diagnose disease like cancer or diabetes or something, to that effect. Other areas are looking at indwelling devices within the oral cavity, leveraging the accessibility of the oral cavity to the rest of the body, targeting different markers in saliva, for instance, to be able to capture different biosignatures relevant to disease areas and of course, the pandemic. But so those are some examples, and so now we're looking at the next phase of this RADx RAD program, and given the of course ongoing public health need, and also as we are moving very fast in this space, we're starting to understand that there's folks that have more susceptibility to developing severe disease, not just from the initial infection, but also even after recovery from the initial disease presentation of COVID. There seems to be, there's still, of course, a lot of unknowns around this. And so this request for information makes part of an ongoing landscape analysis that we're doing in this space. Across a number of different domain areas, including literature reviews, casting a broad net across our federal partners. And this is really the public facing channel that we are putting out to be able to capture information from the community. And this group, it's, I think, very poised to provide this type of feedback. So really be grateful to have some insights. Specifically, we're looking for information around three main categories. The first one is related to the clinical aspects. So looking at understanding what might be some potential biomarkers and risk factors associated with the disease, not just in terms of COVID, but specifically around like severe COVID, and maybe the long COVID, or what we're calling the NIH definition, we call it post-acute sequela of SARS-CoV-2 infection. So one of the challenges in this space in terms of the long-term effects of COVID is that it's so new that it's being called many different things. So there's even at this point, there's no standard or consensus as to the actual terminology that is used to refer to the long-term effects of COVID. And so that's actually a challenge. So we're looking to come up with a better understanding on the natural history and natural biology of the disease. NIH is actually ramping up a whole new effort around the clinical trial, clinical observation studies. It's entitled Recover. And so they have a number of different recovery clinical work cohorts that have been supported, and those projects are ramping up. It's also over $1 billion worth of clinical studies that are aimed to really help us understand the natural history and natural biology of the disease. But those are just starting, so we don't really know what that looks like in terms of PASC at this point. So we know that there is a lot of activity in the ecosystem. There's different clinical studies that are ongoing, being supported, through industry and other, clinical systems. And so, if we're able to get a better understanding on potential biomarkers and signatures and so on, I think that will be very helpful. The other area is understanding what are some available data and resources, infrastructures across the country and maybe even the world, where we might be able to tap into in terms of getting access to data that might be relevant for conducting perhaps data-driven approaches that could really help us crunch and identify trends and patterns and insights that could help drive new science in this space. And lastly, of course, we're looking for potential novel technologies in the context of detection, diagnostics, and a combination of hardware, software type systems, perhaps, that could be helpful, or even high throughput assay or genomic type approaches that could really help identify new targets and have potential to be pivoted to address some of this PASC issue. So that's really where we are, but this is sort of kind of the next generation, kind of like this is a very forward-thinking initiative. We're not essentially kind of limiting ourselves to just the detection or diagnostic aspect of the disease, but we're really looking, beyond that towards like a prognostic So being able to predict the potential susceptibility of individuals to developing these diseases, the PASC aspect and the chronic long-term effects seem to be pretty severe in some cases. But interestingly, at this point, there is even some discrepancy or inconclusive aspects associated with people that are actually developing COVID. In some cases, asymptomatic people that have COVID are presenting with PASC chronic effects in the long term. And so right now, we are not able to make a full determination or association between those people that actually develop COVID, and in most cases severely, to the fact that they might develop PASC. Some do, some don't. So there's still a lot of unknowns, but if we're able to come up with new approaches that we can predict who's going to be more susceptible to these severe diseases, that will be very helpful. So I'll leave it there, and maybe I'll open it up for, I don't know how much time we have, but, I can maybe address some questions.
That was excellent. Thank you so much, Orlando. Very timely and very deep. And yeah, so we have another 15 minutes. We're going till 1:30, and this is a great time for people to ask questions about this NIH RFI with Orlando here, and to all of our other speakers. So I always have questions, but I'll wait. I want to see if anyone else has comments or questions at this point. Masood, while Orlando was speaking, I was thinking of you and your technology.
Hi, Dr. Lopez, if it's okay, I'll contact you regarding some of the ideas that we are developing in University of Utah.
Sure, absolutely. That would be great. My contact information is in the RFI, and do please feel free to actually respond to the RFI via the portal. All the responses are going to be considered and will be very appreciated in terms of the value that they will add to this initiative. So thank you.
Thank you.
Very good. So does anyone else have any questions before I ask mine? Okay, great. Let's get started then. So Deborah, when I was listening to you, and I know that you said you're originally from New York, I think, or at some point you were from New York. And I was looking at your information about, I believe it was commuting by car afterward, where we are now or in the future with COVID. I think of it as being dependent on mass transportation availability. So in some places it could be that people aren't commuting as much because they're not going to work as much. In New York, the traffic is terrible now, and the ridership on mass transportation is like 20% of what it was pre-COVID. And I was shocked the other day. I was dropping my son off at the train. We live in the suburbs. He was going into the city. And I said, gosh, the app must be broken. Everything is off peak. How could it all be off peak? And so they're charging off peak rates to try to get more people to come back and take mass transportation again. So actually, people are going to seem to be driving more because they want to be in their hermetically sealed vehicle as compared to taking mass transportation. So how does that play out in your survey?
Yeah, for sure. Great question, and definitely super topical for New York. But I think, so what I was showing you though, I'll just say again, that was responses comparing pre-COVID to a hypothetical and hopeful future where the virus is no longer a threat. So people hopefully won't feel the need to be in hermetically sealed vehicles. That said, if you check out our PNAS paper, we also, we looked at transit as well, and I didn't put that in the slides. And there, people did, report a lot bigger drop in transit use than they did for car use. So the transit use drop that was reported in our data set, even for this future when COVID is no longer a threat, included both people switching away from using transit, people who previously used transit but maybe purchased a vehicle during this period, and so now they have a car and they're planning to keep using it, as well as increased telecommuting and therefore fewer days per week commuting. And then the other thing that is tricky about thinking about transit with increased, frequent but not everyday telecommuters, is that a lot of transit is priced as for commuters as though has been previously priced as though you're using it every day. So people used to buy like a monthly pass.
Mm-hmm.
And the monthly pass was priced, so it was like a little bit of a discount if you would otherwise have paid every day. But if people are thinking, well, I'm otherwise going 2 or 3 times a week instead of 5 times a week, then that monthly pass becomes becomes not economical. And then that means that every day you're either physically paying the transit fare or you're deciding to get in your car if you have a car at that point. And then that ends up, transit ends up then psychologically feeling expensive, I think, for a lot of people. I mean, especially like from the New York suburbs, it's actually reasonably expensive to buy a round trip ticket to the city. So I don't know, those are my thoughts about transit. I think transit needs to, I and many other observers looking at this situation really think that transit agencies should probably rethink like how they do their pricing and how they do their service, and try to figure out how to provide better service to the people who are their real customers, and not, and that may be a different group of people than it used to be.
Absolutely. I'm really glad to hear. I knew you were thinking about it because you're thinking about everything, but it just wasn't on the charts. But it is an interesting phenomenon, because I really thought the app was broken when it said everything was off peak in both directions all day. I'm like, I've never seen this before, right? And I've been looking at that for many, many decades.
Yeah.
So it's very interesting. Thank you. So yeah, they are rethinking it. And then Masood, I have a question for you. I was looking at your numbers about the 4 to 10% or 6% on the average,
right,
false positivity and negativity, rates for the test. And I was wondering, how does that compare with the tests that we use today for COVID?
Well, the PCR tests are the gold standards, and they have much better false positivity and false negativity rates. Compared to these lateral flow sensors that you can get from CVS and other pharmacies that Abbott has put out, those can be as, their accuracy can be as low as 60-70%, and they're very difficult to read. So anything other than PCR, which is the gold standard of testing, and it tests for the genetic information, and it amplifies the signal, right? So it makes multiple copies of whatever DNA or RNA strand is fine. Everything else is not as good. But if you have a sensor that detects things in 1 minute, and you can keep trying it over and over again, then over time you can see that you're going to improve its false positivity and false negativity rates as you kind of test on yourself or someone else. False positivity is not a problem, because once you detect using this technique, this incomplete technique, I should say, a positive case, then you can refer the person to go and do PCR, right? And that is more readily available than now than before. But false negativity rate is troublesome, because you don't really want to let somebody into, let's say, an arena if the person is positive and you detect the person as negative. So because of that, one can use kind of concept from ergodicity. So you can have like multiple sensors or an array to test in an array and then improve your false negativity rates, or do it over time if you have only one sensor, and the person is doing it on himself or herself. So there's room for improvement, but when you're trying to detect something in 1 minute without signal amplification and things of that sort, you really don't have much room in terms of improving these false rates, false positivity or negativity rates. One can come up with better sensor structure, maybe better aptamer strands, and heat up the sensor a little bit so you can speed up the reaction times. There are a bunch of things that one can do, but that is what we have right now at this point. But I think, if you imagine, forget about the sensor that I have. If you have something on your switch thing that you can basically do this, or when you're going to a grocery shopping or whatever, you can sample the air and kind of semi-sure that everything is okay. That would be great. So that's the idea.
Very interesting. Thank you so much for sharing that. And Orlando, this is why I think you two should get together based on what you just heard to talk about.
Thank you very much for the introduction, and thank you for the opportunity.
Absolutely. Absolutely. And Samson, I actually have a question for you. So when I saw you looking at the confusion matrix, I think we all have confusion matrices we're in every now and then, but the confusion matrix and the viral and the bacterial, I know a number of years ago, I actually became quite sick after I was in China, and I came back to the States and they couldn't diagnose me. It was, it was presenting as viral, then bacterial, then this, then that. I go anaphylactic in the hospital. It was a mess, so they had their own confusion matrix going on at that level with the doctor. So the work that you're doing along with Dr. Pisani in explainable AI and some of these tools and the machine learning and AI algorithms and the convolutional neural networks you're using, it's really impressive. It's wonderful that you're doing this and you did this as an undergraduate, but could you give us a little feel for how it may help in some of these situations? Like they didn't know what to say to me. They were like, okay, we don't really know what you had. Like, seriously? So how do you think these technologies might actually be able to help in the real diagnosis and treatment?
Yeah, so explainable AI is, sort of like a supplement to the radiologist's work. So you can build a very accurate machine learning model to classify different types of X-rays, but just having that black box model isn't really enough to deploy or diagnose real patients, because it's really important to understand what your model is specifically looking at. So that's where explainable AI kind of comes in. Instead of having for scientists to hand label or hand annotate every single X-ray image, explainable AI outputs are sometimes just as accurate as hand labeled annotations, and helps radiologists identify, where the exact locations or regions of pneumonia, for example, or COVID-19 is on a specific medical scan. And that really helps with the radiologist's work in understanding and diagnosing patients quicker.
And treating them appropriately, right, based on what they find out. Well, that's great. Well, it's really exciting what you're doing, and we wish you so much luck in your master's degree now that you're at MIT. I see you already have your little MIT logo on your little Zoom area there. So that's great. So thank you everyone for all your participation today, the researchers for your amazing research, our tax dollars at work, as I like to look at it, and we're delighted that you're using them this way. We're very grateful for NSF and NIH support for all the work that we're doing, the Convergence Accelerator in particular for this COVID Information Commons, so we can do webinars like this. And also the 4 big data hubs work in collaboration, NSF funded big data hubs, to support this program too. And I want to thank my colleagues at the South Hub, the Midwest Hub, and the West Hub, as we're at the Northeast Big Data Hub. So are there any final questions before we go? Okay, wonderful. Well, thank you again, everyone. Thank you so much, Lauren, for another great job, another webinar. Thank you, Benji, for live streaming, Brian for taking notes to write our next blog post on this, and Tia for being here to support us throughout the program.